Good evening, everybody. This is the first journal club of the Advanced Science Academy for 2020 and 21. Thank you all for coming. This should be, I hope, fast-paced and interesting. But the purpose of doing a journal club at all is to communicate science, which means that you need to get used to, you need to get comfortable with being on camera, asking questions. I'm not really the type to actually think that you're asking a stupid question. These things are to talk, to be excited about science, to talk about cool science and bad science. Cal picked a paper that I've been sending to everybody. It is quite a different, I've had quite a different reaction to it. But again, this is about communication. You should feel like you can say anything at any point. All right. Do mute yourself until you want to speak. Go ahead and take advantage of the raise hand function if you want to ask a question, if you don't feel like interrupting. I'm going to interrupt if I feel like I need to. There are some faculty members on here. I saw Mrs. Patel. I saw Miss Lip. It would be nice if you guys were enthusiastic enough about this process to want to take part in it. Because if you don't, it's torture. If you do, it can be a lot of fun. All right, because like if we're ripping apart bad papers, that could be kind of fun. In this case, I am leveling a challenge at you. I dare you to find a problem with this paper. Here we go. Without further ado, this is Cal Braga, class of 2021, and he is going to be sharing his screen and presenting Journal Club number 4, which is Restoring Metabolism of Myeloid Cells Reverses Cognitive Decline in Aging.
Is it working?
It is. You just got to hit present and you're good.
Sweet.
Yeah.
So everybody can see the presentation good and stuff?
Yeah.
Yep.
All right, cool. So the title of this paper is Restoring Metabolism of Myeloid Cells Reverses Cognitive Decline in Aging. Now, when I read that, first two questions are what is myeloid cells and how are they defining aging? So myeloid cells are really just bone marrow cells. That's all you need to know about that. And how they define aging is the development of pro-inflammatory responses contributing to cardiovascular disease, cancer, frailty, and metabolic syndrome. And then something you also need to know just for the figures is that basal respiration means the minimum metabolism rate to support bodily functions. All right, so on to figure 1. So here on figure 1 in A through D, so A, you can see, so on the y-axis is PGE2, which is the hormone that they're really saying is causing aging. And so you can see that in age brains, if you look at the cells, the black is aged, blue is young. In age brains, the PGE2 is almost 3 times as high. And then in figures C and D, they're just showing, so on the x-axis, the vehicle is just the control, and then the 10, 100, and 1000 is the amount of PGE2 that they're testing. And you can see with the more PGE2 there is, the lower that the oxygen consumption rate and the glycolysis is. So obviously more PGE2 means that it's bad.
Yeah, so what is PGE2 though? Do you want to tell them that, give them some background on what that is exactly?
Sure. PGE2 is it's a hormone, and so it kind of just starts showing up when you're older at a much higher rate, and the more of it there is, the more inflammation that starts to show up, which leads to cardiovascular disease, cancer, frailty, and metabolic syndrome.
Why does it lead to inflammation?
Oh, good question. What does PGE stand for?
PGE stands for, let me find it.
It's prostaglandin E2.
Yes, it stands for prostaglandin.
Prostaglandin is a pro-inflammatory signal. It's something that actually is supposed to increase inflammation. That's its one role, right? So we're looking at this, and you have a correlation where you're like, okay, it seems to be upregulated in aging. Why do we care? Right?
Yep. So A through D is kind of just them really just figuring out what the problem is, and they're saying that the problem is the PGE2. And now in E through an E, an F, and then an I, they're kind of figuring out, okay, so how can we fix this? So their first kind of test in E, they take this CD11B Cree, which is CD11B Cree. It's a really kind of crazy thing where once these two things are combined, the CD11B and then the Cree, when they're combined, they allow you to cut out a specific they allow you to just like cut this specific thing out, and what they're cutting out is the EP2, and now the EP2 is the signaling of the PGE2. So by cutting this out, they're essentially not allowing the PGE2 to really do anything. And you can see that when they cut this out in the aged brain, the blue has the PGE2 and it's the aged brain. And you can see that the levels are actually higher than the young brain without the EP2. So essentially what that means, and in F it's more obvious. You can see in the picture the young brain without the EP2 and then the aged brain without cutting out the EP2, huge difference. And now with the young brain without the cutting out the EP2, and then in the aged brain with cutting out the EP2, it looks almost exactly the same as the young brain, which is
okay. So I just want to slow down for a moment. And orient us to what you're looking at, because I do want to give, because this is a journal club. Journal clubs tend to be about like what type of techniques are being used in science that might be interesting. I can explain Cree locks because that's pretty cool. But what you said, there's a big difference. What am I looking at? What are we looking at in F? What is it that's different?
Oh, okay. So F, what the arrows are pointing to are the mitochondria.
Right.
And so you can see the mitochondria in the aged without cutting out the EP2 does not look how it's supposed to look.
Right.
But then the aged with cutting out the EP2, it looks exactly like the young, which
right.
is essentially showing that they can turn the mitochondria back into what looks like young mitochondria,
Yeah.
even though it's aged.
So F is actually a transmission electron micrograph, and they've used a special stain that if in metabolically active mitochondria, it turns dark, right? So since the aged one over on the right, top right, isn't dark, it's not very metabolically active. You guys understand what I mean by metabolically active? Like your metabolism is higher, therefore you're using energy and doing stuff, doing work, and aged cells aren't doing that as well. Kirsten.
Does the cell naturally cut out EP2?
No.
No, it doesn't.
So, Cal, you want me to do that explanation?
Sure.
Okay. All right. Let's talk about cocaine for a moment. Some of you have had neuro with me or whatever, and some of you have taken AP. I tend to talk about cocaine because it's a good model for how receptors work. Okay, so you have prostaglandin E2 is a hormone. That hormone needs to bind to something, a receptor, in order to send the signal to the cell, right? Okay, if you don't have the receptor, that hormone is useless. So an example I can give you is crack babies. I think we've talked about this a lot in a lot of classes. Crack babies have been found to be completely immune to cocaine later on in life because they don't have the receptors for them, right? Mom did a lot of crack while she was pregnant. Babies' receptors got downregulated, so now they have neurons with no receptors for dopamine. So if that baby grows up and then does a line of cocaine or a bag of cocaine, they don't feel anything because they don't have receptors, right? Another example would be the androgen insensitivity syndrome, which I've talked a lot about in most of my classes, where you could be XY, which is male, but you look and have the physiology of a female because your cells don't have a receptor for testosterone. You need both. You need the ball and the catcher's mitt in order to send the signal. Okay. So if I have a ball but no catcher's mitt, no signal can be sent. If I have a catcher's mitt with no ball, no signal can be sent. Okay. There's no passing of information. So Cree locks is a fascinating method where what you do is you take this thing called a LOX P site. It's called floxing. You put a LOX P site on either side of a gene, and so that gene is now floxed. So what they did was they took EP2 in mice and they floxed it. Cool. Okay, fine. What does that do? Nothing at all. Then they had a different mouse, and that different mouse was something that produced a special enzyme called Cree recombinase. And that enzyme was under the control of a different gene. So that whenever CD11B was expressed, you all know what transcription is. Okay. So when CD11B is expressed, the Cree recombinase will also be made in those cells. So what does that do in that animal? Nothing at all. The magic happens when one animal is a mommy and one animal is a daddy, and they love each other very, very much, and they make baby animals, and those babies have now mixed the two genes. So if you have a floxed EP2 in the same cell as your Cree recombinase that you're making, the Cree recombinase goes and kills the gene so that it's gone. So CD11B, Cal, is produced where? Where did we say? I forgot. In myeloid cells, wasn't it? Wasn't it in bone marrow cells?
Yeah.
Specifically? Okay, so that means that EP2 functions everywhere else in the body except in these animals' myeloid cells. So they were able to specifically target a gene and knock it out, but only in the cell type they wanted. That is super powerful. What if you wanted to look at just brain cells? Go find a gene that's only expressed in brains, put the Cree recombinase in that, and that'll knock it out. I've done that a bunch of times, and it's surprising how resilient your physiology is, because sometimes animals will have major genes knocked out and they'll be like perfectly fine. It's pretty, and then other times it's really noticeable. Going back to cocaine again, because why not? Dopamine transporter, which you all kind of know what that is, I think at this point. That is one of the ways to suck up dopamine back into the cell. You can knock that out in a mouse. And if you want to see something funny, look up a video of a DAT knockout mouse, a dopamine transporter knockout, because that thing never, ever stops moving. It's constantly running around like ADHD around the cage, constantly, and it won't stop until it passes out. So that's Cree locks, which is a really, really cool method. All right, go ahead, Cal.
So the next figure that's pretty cool, I think, is look at figure I on the bottom left of the page. And here what they kind of did was they knew that knocking out EP2 helps with oxygen consumption rate and glycolysis, but they figured, well, let's see if it also helps with fighting off viruses. So they introduced E. coli into the experiment, and you can see that in aged ones without the EP2 or with the EP2 not knocked out, it really doesn't fight it too well. But then once you cut out EP2, it starts fighting it better than a young brain, and almost as good as the young brain with knocked out EP2. So there's really just no decline in attacking the E. coli with an aged brain or with the aged cells when you knock out the EP2, which is another just ridiculous finding.
Okay,
So any more questions on figure 1? No? All right.
I have a question. When we get to tear apart this paper?
I'm sorry?
When is the whole critique of the paper? Is that after Cal presents us all the information, or critiquing during?
Whatever you want, Cal. You decide.
Yeah. I mean, if you find holes in it during, then yeah, sure. We can do it right now if you want.
Oh wait, it's more like a statistics thing, and I don't know if everyone's into that. So we'll get you through everything.
Right.
You should go first, and I'll touch.
Sounds good. Now on to figure 2, and in figure 2, they're doing this with mice, and so if you look at C and D, they're testing the mice's memory. And so you can see in C with they did a whole bunch of memory tests, and when they don't cut out the EP2 on the left, they're really all over the place. They're not really in the target quadrant that they want. But then once they cut out the EP2, they're almost only in the target quadrant. So the memory just got like a ton better. And then in D, they just show this in a graph, and you can see that on the left of the graph with the young age, with the young, they spend almost all the time in the quadrants that they want. But then with the age brain in the red without knocking out EP2, they're really not doing too well with a time in quadrant. But then once you knock out EP2, again, they're better than a young brain and almost as good as the young brain with the EP2 knocked out. Same thing in D, except they're just talking about the distance to the target hole. Yeah, Dr. Char?
Yeah, just to give a little background on that. So this particular Barnes maze is that they have to, they'll undergo training a certain day, and there'll usually be some sort of piece of food or treat because they're usually food deprived, which then motivates them to find the food. And so they'll go through a training and they should learn, okay, food is located here. So when Cal says that they spend most of their time where they're supposed to be, that's where the food should be the next day, because that's where it was the day before. But if you look at on the right figure distance to target hole, right, and you see that the red aged group, Marco, that's the one on the left of the aged two, is a lot higher because they're wandering around aimlessly, like they don't know where anything is because they can't remember it because they're old. But once you knock out this particular gene, this receptor, all of a sudden they're not better than young, but they're not significantly different from the young anymore. And that is a functional thing. That's pretty amazing. Before we were just looking at cells. Now we're looking at actual animals behaving. That is marvelous.
And then another kind of functional thing is in figure or in E, they're testing learning, and if you see the like kind of black line, that's how the young brain or the young mice like learns, and then the red line, which is below, that's how the aged brain or the aged mice works when you don't cut out the EP2. But then the green, which is on top, that's the aged mice when you cut out the EP2. So like there's a huge difference in learning too. It's not just memory and like virus fighting. There's also learning applications to cutting out EP2.
Right. This is the cellular version of learning. This is called long-term potentiation. So they kill the mouse, they take out their brains, they look at their hippocampus where you form memory, and they do patch clamp on it. And then they give it a quick pulse, like boop, boop, boop. That's what those 3 arrows are at the bottom. Boop, boop, boop. They give them stimulus, and what happens is the cells get potentiated. Their electrical signal gets higher and stays that way, and that we think is cellular learning. And the red line, which is the bottom one, Marco, they didn't really test. There's a criticism. They didn't really test their figures for colorblind people. I had a friend in graduate school who was colorblind. I always said, "Here, can you read this?"
Nah, I can see that color.
You can? Okay. Because if you can't read it, it's not a good figure. You need to go redo it. But that is the aged one at the bottom. They're not learning their cells aren't even functioning very well, which correlates very tightly with their performance in C.
Yep. So now in I, the last figure, it's a super complicated figure, but like really what they're showing is that this isn't just showing up in mice. It has the same effects in humans, which is like kind of the big thing. Like if you can show it in mice, can you also show it in humans? And they're showing that they can. Any questions on figure 2?
So figure 1 was about PGE, right?
Yeah.
Figure 2 was really about the EP2.
Yeah, they were
So-
they were testing how cutting out the EP2 would affect the mice.
And like this is like you have an old mouse, then you cut out EP2, how it changes then after the fact, right?
Yeah.
No, developmentally, this happens developmentally. So you actually have to age the mice after. This is a long study. I mean, how old? Hold on. How old are aged mice in the study?
They were.
Yep, it says young or 2 to 4 months old, which is really young. I'd almost say those are juvenile. And then aged is 20 to 24 months. That means that what they had to do was they had to wait 2 years of waiting around just for them to get older so they could test them. I love science. You got to be real patient, you know? What if the power had failed or there was a flood and then you lost all those mice? That has happened before. So 2 years they had to age them. And so these mice that had it cut out were Krelox mice. They were aged with it already missing, if that makes sense. RC, yeah.
I have a question for Figure 2E. What's the difference between the cellular like recognition and brain recognition? Because I heard you said that and I didn't know what the difference was.
So the 2000 Nobel Prize was awarded to Eric Kandel, who's at Columbia, because he's the one who actually discovered long-term potentiation. The dirty secret about it is that neuroscientists, we think that it is a cellular model for learning at a bigger scale, right? Where is this published, Cal? This is published in Nature, right?
Yeah.
Nature is the best journal in the world. You can't get into Nature if you just show behavior, or if you just show a cell model, or if you just show the electrophysiology, which is E. You can't get it in Nature. This tour de force did all of that and a bunch of, and then the rest of the paper, which he's going to cover in a moment. But it's we think correlation is not causation, right? We can all agree and nod our heads and bow that yes, okay, we understand that correlation is not causation. However, the correlation between LTP, long-term potentiation, and the mouse's ability to learn things is very, very tight. So we are convinced that it's essentially the reason. It's essentially the way that cells learn about other stuff, and then together they come up with processing as a network, and then that network interacts with other networks in the brain to give you a whole brain output, which is behavior.
For E, is there any significance to like black being higher, like at the very beginning, like than the like non-knockout?
It's not. But now I want you to tell me why it's not, RC.
Like why it's not significant or why it's not higher?
Nope, that's it. That's the answer. It's not, because it's not significantly different.
Yeah, okay.
Yeah. So things may look different. It doesn't mean they are, unless you see that little star. I can't. You're not looking at my screen. See the little star over on the right side of that figure? That little star means significance at p is less than 0.05.
Okay, I didn't realize that was a star.
Yeah, that's what we generally use as asterisks. Every time you see an asterisk, it usually means something significant. All right, Cal, go ahead.
Okay, so and just a kind of a disclaimer. I'm not sure if anybody else noticed it, but when I first went over it, I did. The groups of mice, they might seem kind of small, like they're some of them are 3 and then some of them are 5 or 6, but that's not a problem. That's kind of normal, and it's enough mice for what they're trying to test.
Miss Liff, was that what you were going to ask about statistically?
No, I was going to raise the attention from figure 1, a use of the plus or minus SEM as opposed to standard deviation.
Uh-huh.
I don't know I'm not sure how much into statistics these guys have gone, but standard error of mean helps to narrow your actual statistical, like your whisker bars. It narrows them. So what looks like what you're seeing is a much shortened range as opposed to the standard deviation, which is a deviation from the averaged mean.
Yeah.
And it's much larger. So it
Yeah.
actually helps your paper to, whenever you're writing anything, it helps your data look as though it's presented more in what you're trying to show it.
Yeah, I agree. I had that same problem with my first paper. I wanted to use standard deviation, and my advisor said no, and he wasn't a statistician, but Dr. Gamble, who I talk about a lot, she is a statistician, and my brother is a statistician, and they both said standard error was actually appropriate because it was talking about a population. It's extrapolating to a population rather than just from the sample. So for those of you who have no idea what we're talking about, error bars are absolutely necessary in any type of statistics ever. Okay, so you better never give me anything that doesn't have error bars, as long as you have some sort of average, right?
So like when you guys, if you were to pull up this paper, like the actual paper, and look at the figure 1 caption, almost all of the figure 1 caption is about how they ran the statistics. And that, when you get older into your college years, that is the #1 most important thing about the paper.
Yep.
As opposed to what the paper is saying.
Yep.
The most important thing are your statistics. Second important thing are your graphics. And then
I did not pay Miss Liff off to say something like that. That's actually, she's coming in with an independent voice saying exactly the same thing I tell you guys all the time. What's the most important thing in the paper? The figures. Most important thing in paper. I'll give you that $100 later. All right, continuing. So we can talk more about the SEM thing later, but that type of, that's a great point, Miss Liff, and that's what journal clubs are all about. Like some people have different impressions. You might get statisticians who are going to disagree about whether that's appropriate or not to use SEM, but at least it's something that'll fire people up, you know? Okay. Keep going, Cal. We're on, so figure 1, PGE2, figure 2, EP2, and it also linked behavior to the cellular data and the molecular data. Okay, so now figure 3.
Now in figure 3, they decided to kind of switch it up and use this thing called short hairline GY, or short hairpin GYS1. And that's really just another thing that's blocking the transmission. So they're kind of just testing to see if they can get the same results as they are when they're cutting out the EP2, and they do. You can see in figure B, the columns on the top, like the red ones, those are the aged ones when they're not cutting it out, and you can see that almost everything is upregulated. But then the light blue ones, or no, the dark blue ones right to the left of that is the aged ones when they are cutting out or when they are using the short hairpin GYS1, and it's not exactly the same, but it is very, very close. Almost everything else is downregulated, and there's a huge difference between when they aren't using the GYS1. And so that's really what every single figure in this thing is showing. They just keep on doing like more and more experiments with it, and every single time they're showing that there's a significant difference between age brains when they do use it and when they don't use it, which is like in C again, they're showing that with the age brain it's a lot higher, and then with the age brain with the short hairpin GYS1, it's almost the same level as just a normal young brain.
Okay, can you go back to figure 2 for a moment?
Sure.
And it F, okay? I just want to, do you want to talk about that, or you want me to just mention F?
You can mention it.
All right, look at these things. These are little bands on a gel. You guys are going to run this hopefully over the summer. This is what I really like to teach you how to do. Not this particular experiment. This one is actually called a blot, and it looks at proteins. We're going to look at DNA instead. But look at the bottom row, and it says actin, right? I'm pointing again as if you can see my screen. Actin on the bottom. That is what you call loading control. That's the thing you compare everything else to, all right, as a proportion. And then you ask, then what they're doing is they're asking, is it there or is it not? And so you can see EP2. Is it there in the control mice who just have the Cree? Yes. Is it there in the knockout mice? And you see that it is quite reduced because it's not in their myeloid cells anymore. What you also then see is what's happening downstream. So I'm kind of using an analogy here of baseball. I've got a ball. The ball is your prostaglandin, okay, the hormone. That has to hit the catcher's mitt, which is the receptor. Then what happens though? Like most people would stop the study right after figure 1. Figure 1 would have been the end of the study. You can get a nice paper in Nature in Nature Neuroscience probably, or the Journal of Neuroscience. But they didn't do that. They went on and said, no, we want to know more. What if you've got PG2 going to the receptor? So that's intercellular signaling from one cell to another. What's happening inside the cell? What's the intracellular signaling? And what they found was that AKT, which is a type of kinase, gets downregulated as well in these knockout mice. And AKT is responsible for phosphorylating this thing called GSK3 beta, which is a really cool. I should have asked Dr. Gamble to come to this. So I wrote a review with Dr. Karen Gamble in 2009 about GSK3 beta. GSK3, this is such a cool connection, okay? GSK3 beta is important in circadian rhythms. It seems to be a major target of lithium in bipolar disorder. It seems to affect people who have depression. So it seems to be in this system a major target, which is why they're looking at it. So P, if you're looking at the gel, P GSK3 beta means phosphorylated, and you see that it's less in the knockout. But regular GSK3 beta that's not phosphorylated looks essentially the same. It's fine. Okay. Then there's this GIS1, which is the short hairpin that Cal just mentioned, right? Do you want to explain what that means? What is the SH? What does that mean?
I don't know.
What's the central dogma of biology? Anybody can pipe up. Central dogma.
Sequence equals structure equals function.
That's the mantra. What's the central dogma? What's the best application of that?
DNA to RNA to protein.
Correct. Right. DNA to RNA to protein. Because DNA is simply recipes for proteins. So if I want to see if a protein's having a certain effect, wouldn't it be cool if you could just turn it off? So you can turn it off genetically, which is what they did in figure is it 1 or 2? In figure 2, right? In figure 2B, they turned it off with genetics. They knocked it out. But then they're like, you know what? Let's be really good scientists and test it a different way. What if we can stop the mRNA from working so you can't make a protein from it? And so you use this thing called RNAi, RNA interference. And that's what Cal's talking about. The SHG, GYS1? SHGYS1 is a little piece of RNA that interferes with the mRNA so you can't make a protein with it. Does that make sense? So just another way of showing the same thing. And, he said about figure 2I, that it's the same thing over and over again, and it is. But I want you to appreciate that these scientists did the same experiment different ways to over and over again show you that they're right. Instead of just doing one experiment and going, look, oh, cool result, moving on. They could have published, this is 4 or 5 regular papers and only 1 Nature paper. Okay? Go ahead, Cal. You can go back to 3. Sorry.
Sure. So are there any more questions on figure 3? Here again, they're really just being great scientists and just doing another experiment after another experiment, and they just keep on getting the same exact result. kind of just pounding it into the ground, if you will.
Can I once again I just want to show them how to read, because these things are cool. The journal clubs really are about methods, about learning about different ways so that you in the future will know they exist and can come up with new experiments using those methods. If you don't know the method, you won't know how to solve your problem. But if you look at B, for example, okay, 3B, each one of those little boxes is an individual animal. Okay, and it's specifically each row is a different gene or a different protein, really. And all it's doing is measuring how much of that protein is up or down regulated. So if it's red, it's up regulated. If it's blue, it's cooled off. It's down regulated. And then up on this is a kind of a confusing way to do this, especially if you're colorblind. But the actual animal's genotype is listed up above, and it says aged SCR, aged SHG, GYS, et cetera, and you see how they have a color. So all you have to do is match that color to the top row of boxes, and what you find is that the aged scrambled, SCR means they just scrambled it, and so it's a control. The aged is all red. You have a bunch of upregulation in every one of those things. Anytime you see IL, that's interleukin. Tumor necrosis factor, a TNF. Those are all inflammatory signals. So when you age, naturally, all your inflammatory signals go up. And they are saying there's lots of evidence that inflammation causes dementia. That is called the inflammatory hypothesis of Alzheimer's, right? But right next to them are in the top row, the darker blue boxes. Those are the ones that receive the RNAi, and they look almost just like the younger ones, don't they? Look like much cooler blue. Now, just stop for a moment and ask yourself, why should you care? Is this applicable to you? Do you want to say anything about that, Cal? Like, at this point, what should we be getting from this? Like, why should we care?
Yeah, it's definitely applicable to everybody, because everybody is going to end up aging, and in the future this could turn into like a drug that really would almost reverse aging and kind of bring your cognitive performance back to what it was as a young brain.
I mean, how many of you have family members who have Alzheimer's or had Alzheimer's before they died? My grandmother had it, and if you're an intellectual, it's terrifying to think that you could lose that capacity. And I've been saying all along that you guys are the first immortal generation, and I think this might be your first real go at it, like because of Figure 4. Go ahead, Cal. Figure 4.
Sure. So Figure 4 in A and B, there it's kind of the similar thing to in Figure 3 B, where on the right the right 4 columns is the aged, like just control, and then I should preface this, where in Figure 4 they start using this drug called C52, which is just the actual drug that will be cutting out the EP2. Like C52 is just a code name because
blocking it, not cutting it out. This time it's just a plain old drug blocking it. The way cocaine blocks dopamine transporters, that's all it's doing.
Mm-hmm. So C52 is just a code name for the drug because it hasn't been released yet, so they can't use the actual name because of just patent reasons. So with this C52, you can see that in the darker blue right next to it, the left 3 columns, it's almost everything is downregulated, and it looks again almost the same as the young control and the young with the C52. And then same thing in the hippocampus, which is where you learn. On the right 4 columns are almost, well, everything is upregulated, but then once you introduce C52, which is the dark blue, which is 6 over, almost everything becomes right down regulated and looks the same as the young brain. And E. So, and then in E, they're training mice and then kind of testing what the effects of the C52 are, and you can see that there's a lot of statistical or significant differences where in the preference without the C52 it's a lot lower. There's really no preference, but then once you introduce the C52, which is all the way on the right, is the older mice with the C52. There's a huge difference, and it's almost the same level as the young brain. So I think that kind of, and then like really all of the other figures are again just doing the same experiments, these like more and more experiments with the C52, and every single time they got a significant difference, and that's just them doing more and more experiments and being extremely thorough about it. Does anybody have any more questions on figure 4? That's a no. And now, so why is the screen blank? So now, why do we care? The most important part. So kind of first, what they found was that cognitive decline is possible to reverse by inhibiting the PGE2 signaling. That was what they showed kind of in figure 1, A through D. They showed that like the actual problem was this kind of accumulation of the PGE2 hormone that was then followed by cognitive decline and a slew of problems. And then in figure 2, they just did a ton of experiments showing that an EP2 blockade essentially reestablishes youthful immune system and its effects in both the blood and the brain. So this is kind of saying that with this EP2 blockade, you can almost bring back everything that the useful immune system and the brain can do. So the cognitive decline that's brought on by the PGE2 is essentially cut out, and there's really no decline. And so the big thing is that this can serve as floxing this EP2 will eventually serve as an alternative to anti-inflammatory drugs that are on the market right now that also suppress beneficial signaling pathways. So really it'll be able to do the same things that the anti-inflammatory drugs are doing right now, except you can be more specific with what signaling you're floxing. So that would lower the bad side effects with the drugs that are out right now have. So
one small correction, not the floxing. Floxing is a genetic alteration that you'd have to do before a child was born. What you meant was the C52. Either that C52 or that PF, right? Those were drugs, and they're using code names, we think, because they haven't been released yet, and they're going to be big pharmaceutical sellers. You know, study was done at Stanford. So, first of all, any questions so far for him?
Well, like, does this serve like as a just like general, I guess like, can it just act as like a general anti-inflammatory drug?
Cal, stop sharing your screen for a moment.
Sure.
Can you guys see mine if you pin me and make me big? This is the actual paper, okay? And figure this thing, you need to appreciate that every single one of these little sub-figures is itself an experiment. And so just in the 4, quote unquote, 4 figures, because really should that have counted as 4? You know, like there was 30, what'd you say, Cal? 30 figures or 30 sub-figures, something like that?
Yeah, 2 or 33
experiments in one paper. And then if you had read past 7 pages, they ran all these. This is a tour de force. This is a heck of a study. But I want to draw your attention to this. How do I zoom in? Hold on. I'll do it the old-fashioned way. So this right here is actually the pathway from that PGE2 can be stimulated by. And the main thing that stimulates it is called COX-2. COX-2 it's an enzyme that is itself inhibited by NSAIDs. So the classic NSAIDs are aspirin, ibuprofen, which is Advil and Motrin, naproxen sodium, which is Aleve, meloxicam, Celebrex, which is celecoxib. All of those downregulate this. So can this be used as an alternative therapy for inflammatory disorders? Maybe, right? That's your question, RC, right?
Yeah.
Yeah. So I think so. Sean, were you gonna ask a question?
I was going to ask something along the same lines, like since it works as an anti-inflammatory, would it affect them long term for their physical capabilities? Like would the older mice, since it was cut out at a young age, also be able to move and other things more effectively?
Did they mention that in the paper anywhere?
They kind of did it in figure 3 when they were doing, well, now those are really just memory tests, nothing
Right.
like
But
physical.
nothing physical. So
Yeah.
isn't that kind of an obvious question to ask?
Yeah.
Why didn't they even mention it? Be really cynical here. Why didn't they mention it? Somebody else. Somebody cynical. Sam, you're cynical. What? Why wouldn't they have mentioned that? They're not stupid, obviously. So they know that that's a question worth asking. Why didn't they talk about it? Cynical.
Is it because it doesn't work that way? That's
one possibility, and they don't want to say that, right? Because if they say it that it doesn't work that way, then we're not going to be as interested in it. That's a possibility, but I don't think that's the reason.
Because they don't know.
Oh, I think they do. Who said that?
Ms. Liff.
Yeah, I think they do. No, be cynical. Come on. Why would they do that?
What's the question that you asked?
Why wouldn't they mention that really obvious point that Sean just made? Like, is there a physical aspect to this that is in addition to the mental one?
They want to publish another paper.
Exactly. I bet you serious money they already have the data on it. I mean, this study, just to do the experiments, took at least 2 years. This study cost at least $1 million to do. Okay. And there's lots of people, if you look at the author list. So if I go back over here, let's look at the author list. I almost sent an email to the first author. He's an MD PhD. Paris Minhas, right? Hernandez. There's a lot of people on here, and a lot of them are actually really young, which means that they're working on the same project from a different angle. I say it's cynical because it's like they're going to come back and hit us again with something else, but actually that's the way to play it. They're doing the right thing. Do you guys have any questions or points you want to make about this?
Just one thing. Is there anything done on these in terms of side effects, or do they not publish that in studies like this?
Cal, do you know the answer to that one?
I did not see any side effects.
Right. I don't. This isn't the place for that. That would be in the FDA trials. But the fact that they're code word naming it C52 and PF, and we went looking, we can't find very many other papers about this particular chemical. So we think that it's actually probably under FDA review now. They're just doing, this is like the first level is the animal tests. You got to show that there's major performance gains, right? And if, to Sean's point, if it does affect you physically, that's the ball game. I mean, you got physical and mental, the two main things. What's the third thing? Reproductive. Will it increase your will it put off menopause? That's the third thing worth considering, but it's not like I'm pretty sure that, not to speak for all the females here, but I'm pretty sure that you're good not being fertile forever if you get to live forever, right? Do I have to have a baby every year for 100 years? Like, I don't know if you're really into that. Maybe you are. I mean, most of us are Catholic, so it's kind of required of Catholicism, right?
I think that's the least concerning part of menopause, honestly.
Is that, well, oh, that's a good point. I don't know if they'll know what you're talking about. Can you make that point? What's the most concerning part about menopause that I should have considered, but I'm a guy, so I was stupid and didn't?
There are so many crazy hormonal changes that happen for females, like just total hormone changes. You have weight gain, you have hair loss, change in hormonal activity, hot flashes from hell, all sorts of other side effects aside from just not being fertile and reproductive anymore.
Yeah. So I apologize. Did I offend anyone? That was a good point. I don't know. I'm a guy. I went straight to the making babies part, but yeah, that's a good point. So that third thing really is then maybe you can do that. And you also said something that really appealed to my interest, which was hair loss. So 7 years ago, a paper was published in Nature showing that the gene responsible for male pattern baldness was something called PGE2. So this C52 drug, I mean, I shaved my head for this. I was so excited about this journal club. I did it all up because I'm hoping I won't have to ever do that again once this drug comes out, because I'm going to have a mullet. That's my dream. I need to have a mullet. Okay. Is this in the front, party in the back? True 80s style. All right. So kind of last things. Anybody, do you guys understand how cool this paper is? I thought this is a great one to start with. That was a good job, Cal. This thing is probably the best paper I have read in a decade. They thought of so much stuff. I mean, I couldn't come up with a criticism of it, which is part of the fun of doing journal clubs, is being like, they didn't run this control and that was stupid. Why didn't they go and do this? And why did they went all out and published in the best journal in the world? And then, so that means this guy, what's crazy, Cal, because I looked him up, is we were talking about this yesterday. I was saying to Cal, he's got his career made, except he's an MD PhD and he's going to go practice medicine. So he's not actually going to be doing research. So he's not looking for a faculty position, which I think is a waste of a Nature paper. Give it to one of the PhDs who will actually get a career out of it, you know? But this thing is, if these drugs can specifically target the PGE2 signaling pathway through that receptor EP2, which then signals through glycogen synthase kinase, then you might be able to hit all of these disorders: male pattern baldness, dementia, physical aging, bipolar disorder, circadian rhythms disorders, all of this solved through one small pathway. And I'm more excited about this. This is the thing I'm most excited about since CRISPR came out. So maybe not 10 years, 5 years, okay? Because CRISPR is the way that you're going to have to enable some of this to make sense. You guys don't seem nearly as excited as I am.
Would pharmaceuticals lose a lot of money with this? Because then that means like all the other drugs that would affect different like heart diseases and things like that would go away because of this drug is in play.
Yeah, that's a super cynical way to think about it, but I think it's a worthwhile way to think about it. Is that's what we bald people were thinking when they said there's already a drug on the market that targets this. And then we never heard about it. Why? Because there's all these things that would be negatively affected. All of the bald people that go do Rogaine or any of the Proscar or finasteride, any of those drugs that work on male pattern baldness, they would lose out on that business. But something like this, I mean, this is the fountain of youth we're talking about. This is crazy. And I kind of, Leah Botarelli's not in AMI, right? So she said she gave a presentation last year in which she ended the way I'd like to end this, which is, and we can look like this forever. It was kind of the point, right? I mean, wouldn't it be nice? Wouldn't instructors and faculty, wouldn't it be nice to be 25 again, like back in healthy, capable livers, before Sam tried to kill you? If I could go back to that time, that'd be nice.